So you may have heard of the chemotherapeutic called Taxol, and it's a complicated molecule, and it turns out it's pretty difficult to produce. So Nupur and Jordan are going to tell you today about like a new way to bioengineer it. So anyway, I'll let them tell you about the plan and the great progress that they made.
Hi, so I'm Nupur, and I'm going to be presenting with Jordan today. So this semester we were in the synthetic biology lab, and today we're going to be talking about engineering a system to make a drug called paclitaxel, or as it's commonly known, Taxol, as Dr. Decker introduced it as, and we did this in a soybean plant. So Taxol is a really effective and commercially successful cancer drug. However, the compound that goes into it is only found in the Pacific yew tree, which is pictured here, which is also an endangered plant. So this tree takes about 100 years to grow, and the yield of Taxol that you get from it is actually really low. And so you kind of get this supply issue with it, and that poses a risk to patients' health because there aren't really a lot of effective substitutes for Taxol, and patients do need it. So synthetic biology is a field where we can take parts and abilities from one organism, such as the yew tree here, and we can actually introduce these to other organisms. So we looked at soybeans, and this provides us with a potential solution to the scarcity issue that we see up there with the yew tree. So we can take the enzymes that are used to make Taxol in the yew tree, and then we can introduce them to soybeans. And in comparison to Taxol, they only take about 45 to 60 days to grow. And they would provide a lot more of an efficient way to get this drug.
So here we have part of the Taxol pathway as it appears in the Pacific yew. We start with the precursors of DMAPP and IPP, which are found in all plants, and combine them to create GGPP by using a GGPP synthase enzyme, whose structure you can see here in the ribbon diagram. This GGPP is then converted to taxidiene by taxidiene synthase. Is a yew tree specific enzyme before it is further adjusted by another yew tree enzyme known as T5 alpha OH. And then the taxidiene can be adjusted by a series of enzymes to create the final product, which is the cancer drug Taxol. So now let's look at these pathways in the context of our particular project. In the Pacific yew, most of this pathway happens in the chloroplast, but the taxidiene has to make it out of the chloroplast to be processed in the endoplasmic reticulum into Taxol. The problem with this in soybeans is not only do they not have the taxidiene synthase, but we don't know how taxidiene makes it into the cytosol. How would you fix this problem? The solution that our group used is to exploit the DMAPP and IPP that is found in all plants. We take these compounds and create the pathway outside of the chloroplast. This runs into an immediate problem, though, in that while we do have DMAPP and IPP in the cytoplasm, we don't have a lot of GGPP. To solve this, we add the cytoplasmic Gpp synthase CRTE. Once we built up some GGPP, we can then use the taxidiene synthase we insert into the soybean from the yew tree. This step allows us to create taxidiene in the cytoplasm, sidestepping the need to export it from the chloroplast entirely. Taxidiene in the cytoplasm can then be altered by the enzymes in the endoplasmic reticulum, including T5 alpha OH, to create Taxol.
So this is kind of a big picture overview of what we're trying to do. So our ultimate goal is to put the genes that Jordan was just talking about into soybeans. But as it turns out, you can't just put these genes in soybeans and expect the soybeans to immediately start making the compounds you're interested in. So first, what we did is we obtained the DNA for our genes and we transfected E. coli with them. So this gave us our plasmids containing the genes of interest and the bacterial backbones, along with the sites where they could then be cut by restriction enzymes. However, we still can't just put these plasmids into soybeans and expect to get results. So again, what do? So it turns out there's another bacteria called agrobacterium, and it has the ability to infect plants and actually transfer DNA to them. So what we're doing is we're actually going from E. coli, then into agrobacterium, and then finally into soybeans. Okay, and then again, we're just going to go over everything, big picture, and then we'll go into more detail on a few of the steps later. So first, we looked at the sequences for the genes that we wanted. And then what we did is we actually optimized this DNA for codon usage. So that means that once we had the sequence that we were looking at, we went in and we altered it so that it would have the codons that are best suited for soybeans, which is what we're ultimately trying to produce things in. So then we sent this sequence that was optimized off to a company called Twist, and they were actually able to create this DNA for us. And it's really cool how that works because we were like literally just able to send that off to them, and then they sent us back what we needed. And then we got this DNA, we inserted it into plasmids, and we transfected E. coli with it. To make sure that we were getting plasmid uptake, then we selected for antibiotic resistance, which we're going to talk about a little bit later. Then we chose the colonies that took up the plasmids, we grew them, we digested DNA, and then ran gel electrophoresis to check the length of the fragments we got to see if we were getting what we wanted. And then finally, we sequenced our DNA to make sure, again, that what we ended up with was what we had originally intended when we designed our sequences.
So here you can see the vectors that we assembled. We first created a GGPP synthase plasmid to increase cytoplasmic concentration of the precursor GGPP. Then we created a taxodiene synthase plasmid to add in taxodiene synthase into the cytoplasm. Since taxodiene synthase is unstable in the cytoplasm, we attached a maltose binding protein, or MBP, to it for stability. We also made a plasmid that combines those two plasmids to create GGPP and convert it into taxodiene in one step instead of two. We also created a plasmid with T5 alpha OH, which is in the endoplasmic reticulum and would hopefully boost Taxol yield.
Okay, so as you could see on that previous slide, there are a lot of enzymes and steps involved in producing taxodiene. So what we needed was a way to create all the tools that we needed and then be able to mix and match them later for various purposes and experiments. So we did this using a method called Golden Gate assembly. So with Golden Gate, we have the site for the enzyme to cut on the gene that we made. So for example, here we have taxodiene synthase. So in the picture, we have the letters representing different sites, and the scissors are showing where we can actually make cuts with enzymes. So then we can take this gene and we can place it into a backbone with additional cut sites. Then we can go in and we can actually add sites onto the ends of pieces that we want to include in a final vector. And we can do that in a way that the vector is in an order that we want it to be in and kind of keep increasing what goes into a piece. So here's an example, but basically by using this, we're creating this toolkit, which you can see in this orange circle here. And you get a bunch of pieces that then you can fit together like jigsaw puzzles based on whether they have an A, B, or C at the end of them. So, yeah.
So to determine which E. coli were transfected and which ones weren't, we used a blue-white screen. What this did is our transfected colonies, when grown on special media, appear white. We prevent any untransformed E. coli from growing altogether by growing them on antibiotics. And as you can see in figure 2, we had a high rate of successful transformations. And after we've done the construction and transfection of plasmids, we want to make sure that we got the right things. So we take some of the restriction enzymes that we discussed earlier and use them to cut our transformed plasmids and run them on a gel. So here we have the CRTE GGPP synthase plasmid, which cuts into 2 fragments, one with a promoter, CRTE, and a terminator, and another with a chlorophenicol resistance backbone. We also digested the fusion protein of CRTE taxodiene synthase in a stabilizing maltose binding protein, which when cut comes out as a large piece with the additional piece of the backbone of ampicillin resistance. And as you can see on the gel here, the cut pieces from the 2 plasmids form 2 distinct bands for each of the plasmids at the correct sizes.
Okay, so this is similar to what Jordan just talked about, but what we're looking for here again is that the lengths on the gel that are cutting into the pieces that kind of reflect the sizes of the backbone in the gene. So on the left, we have T5 alpha hydroxylase. So this is the next enzyme involved in the Taxol pathway once we have taxodiene. And so you can kind of see that the bands here on the gel are where you would expect them to have cut based off of the vector, which you can see on the left. And then on the right is the fusion taxodiene synthase, and that also has the bands that you would expect. So that kind of shows some success here initially. Okay, So just to kind of go over everything that we've done, we've finished this first step of transfecting E. coli, and we've also, so we saw the success in that in our gels, and we've also sent off our DNA for sequencing, and we've seen some initial success with that as well. So looking forward, we're looking to get the rest of our sequencing results back, again, just to make sure that we have exactly what we want to be working with going forward, and to ensure that we didn't get many errors while we were doing this cloning. And then after that, we're going to go into agrobacterium, and then eventually we'll go into soybeans. And then we're going to use gas chromatography, mass spectrometry, in order to see if we've actually produced taxidiene in the soybeans.
Okay, so we would like to thank our fellow group members, Simon, Professor DeDecker, and the department, with a special shout out to Bo. Thank you. And are there any questions?
Is Bo the dog?
Yes.
Bo is the dog.
Yeah, so I have a question, Jordan, and that is, will this eventually go into the germline of the soybean plants that you're transfecting, so that you'll have a sustainable crop to go ahead and harvest this?
Yes, that is the ideal scenario. Agrobacterium is around a 10% efficiency, but hopefully when it does transfect the cell, it will be a germline cell, so you can grow up a whole crop, as you said. And then we get the soybeans from those and we place them under special conditions, so that way they can create the Taxol for us.
Thank you.
Stephen, go ahead and ask your question.
Thanks. So I just had a question about the Golden Gate technique. It seemed like you were saying that you were like adding on consecutively these segments into the same plasmid. I was wondering if you could kind of clarify how the engineering on that works.
Yeah, so it kind of took us a while to understand this as well, but basically what you can do is these sites that say A, B, B, C, C, D, there's sites that you can add on to the ends of pieces. So for example, a site that is like originally AB, if you wanted to add a C onto the end of it so that then you could like attach on a CD piece to it instead of like a different like BC piece, you would be able to do that. So you can kind of like change what you're working with to keep lengthening that. Does that help?
I guess, yeah. So is like A, B, C, and D, are those like restriction sites, or are they like complementary sequences?
In a way, they're sort of both. The special thing that makes Golden Gate just different from a lot of different restriction enzymes is that the cut site isn't exactly the same as the part of it that matches up to the section that you want to ligate together. And that makes it a lot more flexible. So the sites are actually the parts that will match together, but they also have to have a cut site that's right next to them. So it's sort of both.
Okay, great. Thank you.
And it looks like there's a question in the chat. Seeing as IPP and DMAPP are isomeric metabolites in the HMG reductase pathway, would a transgenic animal be useful in this type of synthesis as an alternative?
It's theoretically possible. It wasn't in the scope of our research to use animals, since plants are a lot more harvestable and sustainable because you can grow a field of soybeans, but it's a lot harder to grow a field of chickens. So it's more feasible economically to go about this using plants.
So I have a quick question then. Do you have any idea what size of field you would need to kind of get the amount of output that would be useful for you?
I don't think that we do. Jordan, you can, if you have any idea, you can definitely jump in. But I think, again, like, since we were kind of focusing on creating like one of the first intermediates in the pathway to ultimately produce Taxol, I don't think that was quite a consideration we had gotten far enough ahead to thinking to.
Okay, I think at this time we'll close out the talks. Thank you all so much. Speakers were amazing. And I think it probably is clear, but I just, speaking for all of the instructors, I wanted to let you all know how incredibly proud we are of the work that you generated this semester. It was a challenging semester, and yet you generated an enormous amount of beautiful data, and the presentations were really incredible, both the oral presentations and the posters. So at this time, what we'd like to do is recognize the poster winners from each of the courses. So Dr. Philman is going to share her screen, and if we could ask Dr. Guild to present the awards.